If you look at living things, we can characterize them based on size. We start with California redwood, right? Huge, 100-meter diameter. If you look at the picture, you'll be amazed. A car, a big SUV can drive through. They made a hole in its stem. You can drive through. It's like a tunnel, a short tunnel. You keep on going from animal kingdom, human, birds, come down to our interest, nanotechnology, 100 nanometer, which is the range of electron microscope. So you see light microscope and electron microscope has overlap in that, that region, right? Going from 100 micrometer to uh, a few hundred nanometers. I can say that you can see easily with the microscope up to 3 micrometers with an optical microscope, but below that, you definitely have to go electron microscope. I highlighted here DNA because when you would do uh, come up with ideas, research, you will see that many times there is mutation involved. And when you say mutation, it's a genetic level defect which causes a disease. Right? When that happens, so from 2 nanometer DNA, where I highlighted DNA, it affects things at 1 micrometer chloroplast. That kind of chloroplast course is in plants, but that's a size scale of things we'll look at up to 10, 30 micrometer cells. So think about it like a I don't know, a car. So we drive a car. A car would drive all good until we get into a trouble and you open the bonnet and you find out it's one small part which is not functioning, and that's making the whole car not non-functional. This is exactly what you can think of analogy of cells and their subparts. So when you talk about cell, there are two types of cells. Bear with me, this is nothing more than high school biology. But why we have to go through it? Because when you talk about bionano and you especially look at GRP, some research ideas, you need to know what are you talking about or what are the things that are important for biology. We have two types of cells. We have prokaryotic cells and eukaryotic cells. Prokaryotic, what we know as uh, individuals that are very much contained and they have plasma membrane and they have things in it, kind of a whole mush is all in it, right? Example, bacteria. Eukaryotic cells, they have a little bit more complex structure to it. They have a nucleus, well-defined nucleus. So a fortress within the fortress. So a smaller kind of a thing which contains much more important things in it than the cell. So, and they talk to each other. When I say talk to each other, they sell, they send chemicals. So those chemicals go travel and they go from one cell to other cell. The other cell, once it receives that chemical, it knows what's going on. So this is kind of what we would be looking at, but it doesn't mean they are separate from each other. Bacteria, they come in contact with living human cells and they cause disease, right? So they go back and forth. We know lots of different bacteria. We know virus. Virus is smaller. Virus is not a cell, by the way. So we have to know this distinction: bacteria and cell and virus. Two different things, two different operations. All right. So when you look at a prokaryotic cell, we say everything is all mushy mixed up inside it, right? We have a boundary cell wall. We'll look at that also. What is it made up of? And has a bunch of information. But there are things, all the things that are in there have a certain role to play, right? You should be able to maybe write one line or know at least one liner of what they do. But I want to move to animal cells instead of that. So now animal cell, we have a kind of an order in there where we have a cell wall, we have nucleus, and if you see, there are these small holes on the nucleus also, which is used to send things in and out of nucleus. We have mitochondria, we have endoplasmic reticulum, Golgi apparatus. So each of those things have certain jobs to do. I will not go into all of them. I'll just go through few of them where I which is very high level, where lots of bio nano interface work has been done, right? So when we talk about cell, there is a whole bunch of engineering happening inside. There is a lot of activity going inside, which has one thing affects the other, the second thing affects the other, and there is a whole chain of things. It's amazing to see how much engineering happens within a cell. That's why when something breaks down, for us, it's easy to find out what is not working. So it's made of water mostly, and they are suspended in it. The cell wall, that's the most important thing. It's made up of fluidic kind of thing. We call it a plasma membrane. What it has is, if you look at this one molecule, this red with two yellow legs, that's the fundamental unit that makes up this whole thing. In that red head and two yellow things, the red one is hydrophilic and the yellow ones are hydrophobic. You know, hydrophilic, what does it mean?
It's attracted to water.
It likes water, right? It and what is hydrophobic? It does not
Repels.
like water, repels. So when you throw these bunch of things in water, after some time they would form this wall where red things would be facing outward because they're okay with water, and they'll protect the yellow things because all of them don't like water. Hence, it forms a wall. So that's what the wall of a cell is made up of. But now, then, in that wall, there are a bunch of things that are suspended. They all are protein. We generally they all are called protein because they are made up of amino acids. Some of them are meant to let things pass through, what is called protein channel here. So it's nothing like but just a tunnel for things to go inside or from outside to inside, both ways. It can do very specific kind of transport. There are some things which may be just protecting the wall and making it strong. There may be other things where when something binds on this side, there's some message goes in there and some activity happens on the other side of the wall. So kind of the doorbell, you push the button at the outside the house and a bell rings inside the house. Make sense? The analogy. So there are those things where things don't cross over, but one side interaction would result into other side some activity. So uh, then there are things it may create and give out into the blood. So a bunch of things would happen. Now, if any of those things break down, disease. I give an example. Okay, when we are sick, when we are tired, we take a Tylenol, right? When we are sore, we take a Tylenol. What does Tylenol do? That compound chemical goes to the cells of our wherever it's aching. It goes to the whole body, by the way, and it gives that chemical to that cell. The cell relaxes instead of being in strain. It relaxes. That's a simple process. It does over the whole body, but the whole body where you ache more, there we feel more difference. But that's as simple as that. This cell wall is responsible for that chemical to get inside the cell or to let whatever happens inside to make sure that happens. All right. So these are called phospholipids, these red and yellow things. We call them phospholipid, right? These are fatty acid. Acid means it has OH group in it and it's bound to glycerol. Don't worry about details, but just remember there are two groups. There is head, which is hydrophilic, and there is tail, which is hydrophobic, right? And if you look at it, even it's just basic chemistry. There's nothing to it, just basic chemistry. Carbon, hydrogen, nitrogen, all known stuff, right? But we were scared in high school from biology because they were talking at very high level, right? But today we see this is all nanotechnology.
I'm still scared of it now. Shit.
Yeah, and that's why I'm saying just remember there is hydrophobic part and there is hydrophilic part, right? But then at this stage, I want you to focus more on research and think about it. Okay, what are the problems and how do you break down and understand those problems so we can solve them, some of them, right? So another artistic depiction.